Okay guys, so yesterday at the end of class I asked you to write down some questions or lingering concerns that you had about the content for the test tomorrow. So I wanted to go through some of those questions and try to make sure we had answers to those. So the first question was, what does atomic mass mean? And so mass is really a term that can generally be used interchangeably with the word weight. So just like your weight is how much you weigh, the weight or the mass of an atom is how much it weighs. Now, when we're looking at the mass of an atom, we don't really count the electrons because they're so very small, their weight is insignificant. So it would be kind of like counting a single hair on your head and how much it contributes to your overall weight. It really doesn't, so we don't count it. So then the rest of the atomic mass or the atomic weight comes from the weight of the protons and the neutrons. Now when we look at protons and neutrons, remember those are found in the nucleus of an atom, so they're kind of the core part of the atom, and it makes sense that they have the most weight. Each one of them weighs one atomic mass unit, so you can think of that is the unit, like the unit you would use for your weight would be pounds. Here we're using atomic mass unit instead. And so each proton weighs 1 and each neutron weighs 1. So all we have to do is count the number of protons and the number of neutrons, and that's going to give us our overall atomic mass. And so when we look at one of these periodic table entries, the atomic net mass is always going to be the larger of the two numbers that are provided. And so you can see it here. It's called atomic weight on one of these examples and atomic mass on the other. The other number that we see, this was not one of the questions, but just as a quick review, is the atomic number, which tells us the number of protons. So if we go back to our little equation up here, we see that protons plus neutrons equals atomic mass or atomic weight. And in a periodic table entry, we're given the number of protons here and the atomic mass down here. And so it's very easy then to figure out what the number of neutrons are simply by subtracting the protons from the atomic mass. And that's really how we use the atomic mass whenever we're doing a problem with atoms in chemistry. So the next question that I got was, how can you tell how electronegative an atom is? And this is one of those questions that would be much more complex if you were in a chemistry class. And you can see in the diagram below that there's a trend in the periodic table that as you go from left to right, the atoms become more electronegative. And as you go from the bottom of the periodic table up towards the top, they become more electronegative. And so overall, we can go from the bottom left to the top right, and we can see that there's an increasing electronegativity as we go in that general direction. However, in biology, remember we are only concerned with 6 basic atoms: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And so of those 6 atoms, 4 of them all have electronegativities between 2.2 and 2.6, which are essentially the same when we're looking at that tug of war that can occur between atoms. So carbon, hydrogen, phosphorus, and sulfur, when they form bonds with each other, they're going to be relatively equal in terms of their attraction for electrons, which means they're going to be equal in terms of their pull on those electrons in that tug of war. However, oxygen and nitrogen both have electronegativities over 3, which means that they are going to have a much stronger attraction for those electrons and tend to pull on them a little bit harder. So they're going to be the Incredible Hulk in that tug of war as compared to Shaggy from Scooby-Doo, if you remember that image that we saw in class. So in terms of our biological molecules, it's much easier to remember that oxygen and nitrogen are the most electronegative, and all of those other atoms that we'll find in our molecules are going to be relatively equal, and so they're not going to form those polar covalent bonds that we talked about. And so then that takes us to the next question, which is, how do you tell the difference between the types of bonds? And so remember, these bonds are all about what's happening to the electrons in the valence shell. And so in an ionic molecule, or an ionic compound, what we see is that electrons are transferred. Okay, so remember, ionic bonds, we tend to see an electron transfer. As a result of that transfer of electrons, and so you can see here these X's represent the 2 electrons. One of those electrons came from here, from this atom. The other came from this atom. And so this pair of electrons is completely transferring ownership to this larger atom, which makes it negative. So electrons have a negative charge, so if the electrons go to atom B, then atom B becomes negative. Atom A in this pair loses an electron, and so because it's losing one of those negative charges, it becomes unbalanced in its charge, and it becomes more positive. That positive charge and that negative charge are going to be attracted to each other, and that is what causes the ionic bond. It's kind of like in moving those electrons, we turn those atoms into magnets, and now they want to stick together. Now, the other type of bond that we talked about is a covalent bond, and in a covalent bond, electrons are being shared. So the difference between then a nonpolar covalent bond and a polar covalent bond is in how they get shared. So in a nonpolar covalent bond, we see that electrons are shared equally. So you can see in this diagram, this would be atom A, this would be atom B, and we can see that they overlap here in a region that is very even. And we see that the two electrons are situated directly in the middle of that region, indicating that the electrons are spending an equal amount of time with atom A and atom B. So think back to what we just saw in terms of electronegativity. If we see carbon, hydrogen, phosphorus, or sulfur engaged in a covalent bond with each other, it's going to be nonpolar because those electrons are going to be shared relatively equally between those atoms that have relatively equal electronegativity. However, when we see oxygen or nitrogen involved, because oxygen or nitrogen have a stronger attraction for electrons, when those electrons are being shared, we see that the electrons spend more of their time skewed towards one of those atoms. So if you notice here, the electrons are no longer situated in the middle, they're situated more towards atom B, which means that atom B takes on a partial negative charge. Again, those negatively charged electrons spend more time here, so this atom becomes more negative. Atom A, because it's spending less time with those electrons, becomes slightly positive. And so this is a polar covalent bond. Those electrons are being shared unequally because they're skewed more towards the electronegative atom. And so as a result, the atoms in the bond take on partial positive and partial negative charges. So those are our three major types of bonds that we see. The other thing that we looked at is called a hydrogen bond, but it's really more of an attraction. And so what we see here is that there is a water molecule held together by polar covalent bonds. So our oxygen has a partial negative charge. We have another water molecule over here held together by polar covalent bonds, and so our hydrogen has a partial positive charge. Now, this partial positive charge and this partial negative charge are going to be attracted to each other because they are opposite charges. However, they're very weak in that attraction, so it doesn't necessarily hold those two molecules together for any period of time. That attraction is formed and broken very frequently, and so it's considered a very weak bond or a weak attraction. And so I kind of came up with a flow chart to help us as we work through identifying, is there a polar covalent bond, or is it a nonpolar covalent bond, or is it ionic, or is it a hydrogen bond? And so when you're looking at an atom, the first thing you want to look at is, are there any charges that are shown on those atoms? Okay, if the answer is no, then it's going to take us down this path. It must be a covalent bond if there are no charges, meaning that there's electron sharing. And we probably are going to see that those atoms are connected by a line or by a pair of dots in order to indicate that there's a covalent bond. So then we want to ask ourselves, is oxygen or nitrogen involved? Because remember, oxygen and nitrogen are very electronegative, and so if they're involved, then that would make it a polar covalent bond. If they're not involved, then we probably see some carbons and hydrogens, maybe phosphorus or sulfur, and so that would indicate a nonpolar covalent bond. Now, if we answer that yes, there are charges on the atoms, then it could indicate that we have an ionic bond, but it could also be a hydrogen bond or possibly even a polar covalent bond if the charges that are shown are partial charges. So we want to determine, is there a line or dots connecting those atoms? Because that would indicate that there is a covalent bond present where electrons are being shared. If the answer is yes, then we would see that that's a polar covalent bond. If we have a pair of dots or if we have a line connecting those atoms. However, if we don't see that, then it could be a hydrogen bond where we have partial charges indicated by that sigma symbol on different atoms. And so one thing that I want to point out that I just realized is that we may sometimes see a hydrogen bond with dots connecting it, but it'll look much more like we see back here. Here these are dashes, where you might have dots or dashes connecting those two things that have opposite charges, but the key is that they're partial charges on different atoms that are connected to each other. If we don't see that, if we see that there are full charges on different atoms, then that indicates that it's an ionic bond. Okay, so hopefully this will help you some as you're going through the process of trying to determine what kind of bond might be present. Okay, so the next question would be when would a double bond take place and why would that happen? And so remember, a double bond is always going to be a covalent bond, and it means that two pairs of electrons are being shared. And so it can occur when two atoms have both, they both have at least two unpaired valence electrons. So for example, hydrogen only has one unpaired valence electron, so it can only participate in one bond, which means it's not going to form a double bond. Okay, so carbon has 4 unpaired valence electrons. Oxygen has 2 unpaired valence electrons. So either one of those can participate in double bonds, and we can see that down below. Oxygen forms a double bond with itself to make the oxygen gas that you breathe in. But we can also see that carbon forms a double bond with itself, and then it also has some single bonds with hydrogens at the same time. So these atoms could form double bonds or they could form single bonds, and so it really kind of depends on the environmental conditions as to which one's going to happen. So it's not something that I would expect you to predict, but I would expect for you to be able to indicate that a double bond is present based on the structure. One question asked about the difference between Bohr models and Lewis dot structures. And so you can see here, this is a Bohr model of a carbon atom, and you can see that it shows all the protons and neutrons that we find in the nucleus, and it also shows the electrons in every shell of the outer shells of the atom, so it's got not only the valence shell or the outer shell, but it's also got any inner shells that might be present in the Bohr model. However, when we look at the Lewis dot structure, it's only going to show the symbol for the element and then also the valence electrons that are present. So carbon only has 4 electrons in its valence shell, and so we see only those 4 electrons present around that symbol for carbon. Another question asked, what exactly is ATP? And so ATP is just a molecule. It's a modified nucleotide, and remember, nucleotides form our DNA and our RNA, but it's a modified nucleotide that has 3 phosphate groups. So adenosine triphosphate, it has 3 phosphate groups. And it is just a molecule, but it stores a tremendous amount of energy in its chemical bonds, which is why it's so important in living things, because it's the molecule that provides energy to perform various types of work that we do in our cells. And so I like for you to think about ATP as what we call the currency of the cell in terms of energy. And so currency very often is associated with money. So if you look at the example over here, we spend money in the form of dollars. And think about it as if you don't have a debit card, as if you're actually using hard money. So we spend money in the form of dollars, but if we were to go to another country, like if we went to Europe, we would have to cash our dollars in for euros because that's what they spend in that country. And then we could use that money to be able to spend on our vacation to have a good time. However, we have to make that change from dollars to euros to be able to do that. The same thing happens when we process food. We've got plenty of energy found in our food, and we have to turn that food into ATP to be able to actually spend that energy to do work like muscle contractions in our cells. Okay, so another question was, what is an ester linkage and how does it work? So an ester linkage is just the name of the bond that we see forming between parts of a lipid. And specifically, an ester linkage forms between an alcohol group and a carboxyl group. That's not necessarily something that you need to know, but because we have a fatty acid that joins with this glycerol, that's exactly what happens. So this is a dehydration synthesis reaction that forms this linkage, and then it's what allows this fatty acid to be attached to the glycerol. All it is the name of that covalent bond that forms between the parts of a lipid. The next question was, which part of a phospholipid is polar and which part is nonpolar? So when we look at a phospholipid, remember it also has a glycerol backbone, and then it's going to have 2 fatty acid tails instead of 3. And in the spot of the 3rd fatty acid tail, rather than having a tail there, we see that there is a phosphate group. Now if we look at this phosphate group, it's got a phosphorus and then it's got several oxygens around it, and we can see that this oxygen has a negative charge on it. Excuse me, and remember those oxygens are going to make it very polar. So what we see is that the phosphate head of the phospholipid, because of that phosphate group, is going to be charged and therefore polar or hydrophilic. The tails are only made up of carbon and hydrogen, which form nonpolar covalent bonds, and so this part is hydrophobic and tends to stay away from water. Another question asked which part of the protein is the carboxyl group. And so remember, proteins are made up of monomers called amino acids. And every amino acid is going to have an amino group, which we can see here. Amino groups have nitrogen in them. Also a carboxyl group. And this is the carboxyl group, which is a COOH. And so it's going to be the part that turns it into an acid. We also see that all amino acids have a hydrogen and a variable group or a side chain or an R group, and all 4 of those things are attached to this central carbon. So this is your basic amino acid structure. This part, the carbon with the double bonded oxygen and the OH, that is what your actual carboxyl group is. One of our last questions here, what are the 3 parts of a nucleic acid monomer? So remember, the nucleic acid monomer is called a nucleotide, and the 3 parts are that it contains a 5-carbon sugar, and so that 5-carbon sugar in DNA is called deoxyribose, but in RNA it is called ribose. It's also going to contain a phosphate group, which we can see here. This is going to be very similar in structure to the phosphate group that we just saw in our phospholipids. And then a nitrogen-containing base, and that nitrogen-containing base is where our A's, T's, and G's come from, and we're going to talk about how they pair with each other a little bit further down the road. So if we look here, this is a simplified diagram. Sugar, phosphate, that forms the backbone of our DNA strand, and then our bases are going to be what sticks out and forms kind of the rungs of the ladder where you actually put your feet whenever you're climbing a ladder, if we think about it that way. And then what is the function of nucleic acids? So nucleic acids in general are going to be used to either store or transmit genetic information. So DNA stores the information for every single one of our genes in the nucleus. And so you can think about it as like your master blueprint for the cell. Tells the cell how to do all of the jobs that the cell could possibly do. But then RNA carries the information from one gene at a time into the cell and uses it to build a protein, and that protein, remember, action verbs or action molecules in the cell, or yes, action molecules in the cell, they're going to be the parts of the cell that actually do things that give us traits. And so the RNA transmits that information so that the protein can actually be built to give us a trait. And so you can think about it as just a smaller part of the blueprint or a photocopy of the blueprint for just the part that we need at that particular point in time. A few other questions that were asked that maybe don't require a whole slide. One question was everything past atoms, which I don't have time to do in a quick little video here, but do keep in mind that we have recorded videos available for every single one of the chapters, and you can go in and watch those as many times as you want. You can rewind them, you can pause them, and if there are things that are still troubling after you've watched those, there are crash course videos that you can watch as well that will help to reinforce some of these ideas, and then you can always send me emails with specific questions. One question was, will we have to draw structures on the test? No, not specific structures. I would be familiar with how to draw a Bohr model and what that's going to look like, but you're not going to be asked to draw, you know, a amino acid, for example, nothing like that. The question, at what temperature is water most dense? This is one of those properties of water that we talked about, most dense at 4 degrees Celsius, but water freezes at 0 degrees Celsius. And so unlike many other substances, it's not its most dense in its solid form. And then the very last question, will we have to know each different type of carbohydrate, lipid, protein, and nucleic acid for the test? Yes, you really should. You know, we talked about carbohydrates, we talked about sugars, and then polysaccharides. And in the polysaccharides, we talked about starch and cellulose as plant polysaccharides, and then we talked about glycogen and chitin as animal polysaccharides. We talked about different types of lipids, so fats, phospholipids, and steroids. For proteins, we didn't necessarily talk about any specific proteins, but we did talk about a lot of specific functions of proteins, and I would be familiar with all of those. And then for nucleic acids, you do need to know the difference between DNA and RNA. So hopefully this was helpful. If it stirred up any new questions, feel free to send me an email. I'll look forward to seeing everybody in class tomorrow, and good luck studying. Thanks.